Hello everyone. Today we will be discussing Skeleton Practical Examination Question Paper Pattern Semester 2 FYBSC Practical Paper 2. So time is 2 hours, marks are 50, and how the question pattern or question distribution is that we are going to see now. So question number 1 is estimate vitamin C from the given sample. So the source of vitamin C, as you all know, lemon and all. So two samples will be given to you and you have to estimate which sample has vitamin C in it. So you have to perform that experiment, or you will get, there is another option, estimate maltose content from bread. So that using Benedict's reagent we determine whether the maltose with maltose content of the bread we determine it is a qualitative test. Vitamin C presence is also a qualitative test, it is not a quantitative. We are not going to measure anything. Estimate protein content from two different types of eggs. If the experiment is there, has been taught to you, then okay, then you can prepare for that, otherwise you can keep it for option. So it is this question number 1 is for 15 marks and it is a major question. So it is for 15 marks and it is a major question. So either you will get vitamin C, or you will get maltose, or you will get protein content. So for this purpose you are supposed to write aim, requirement, principle, whatever is the observation, and your results, and you are supposed to perform the experiment. So this is about question number 1. Now question number 2, analyze the given food sample and identify the food adulterants. Any two samples will be given to you, sugar or jaggery or ghee. Generally jaggery or ghee, these two samples are given and you have to analyze what are the adulterants in it. So the tests are there, so you have to remember the test and you have to perform. So if the experiment has been done in the laboratory in the regular practicals, you have to perform that. Or you will get MBRT, that is methylene blue reduction test, in which you take particular amount of milk into the test tube and you add methylene blue, you put a cotton plug and you keep it in the incubator, and after a certain interval of time you see the color change, right? So you have to evaluate the quality of milk by MBRT test, that is methylene blue reduction test, MBRT method. So the alternative to food adulterants is MBRT, or third alternative to MBRT is determine efficacy of different antacids like gelocil or eno or anything. So any two samples will be given to you and you have to test which is the efficient antacid, which is the best antacid, that you have to remember. So determine the efficacy of different antacid is the third alternative. So analyze a given food sample and identify food adulterants, or you will get MBRT, or you will get efficacy of antacids. This is for 10 marks, it is a minor question. Now, and you have to, if the examiner asks you to write aim, requirement, principle, you are supposed to write it, observation, results and all these things. Question number 3, determine the moisture content from the biscuit. It is very easy experiment. You have to just weigh the petri plate in the beginning. You have to put the biscuit into it, take the weight of the biscuit after putting it in petri plate. Then you have to keep it in oven and you have to afterwards you have to subtract it and you have to determine what was the moisture content. So it is very easy experiment, minor experiment, just for 5 marks. The second alternative for that is question number 3, alternative for moisture content is, on the basis of microscopic structure you have to draw the structure of the starch granules. You have to study the microscopic structure of the starch granules. Any two cereals will be given to you. So it is a microscopic examination of starch granules of cereals. Or you will get adulterants from present in the milk, urea or any kind of any adulterants if it is present in the milk. So you have to detect the presence of adulterant which is present in the milk. So you have to detect that. Okay, so these are the 3 options with you. And fourth option is there, determine whether given blood sample from the anemic or non-anemic person using you have to determine whether the person is anemic or non-anemic using a copper sulfate solution. A copper sulfate solution will be provided to you into the coupling jar. You have to put a drop of blood, and if that drop of blood sinks, or if it floats, based on the results you have to determine whether it is from a anemic person or a non-anemic person, and you have to suggest a proper diet to that person if it is anemic. So these are four alternatives available in question number 3. Anyone you can get in the practical exam. Question number 2, 3 alternatives are there. Question number 2, there are 3 alternatives. So all are the activity experiment. Question number 4 is your identification, which is very important and which is for 10 marks. So now identification, you will have 5 spots A to E, and first spot will be protozoan. So A में आपको protozoan ही दिखाया जाएगा. So it could be either entamoeba or it could be either plasmodium. Entamoeba या plasmodium कुछ भी एक हो सकता है. One specimen from helminth parasite, ascaris या wuchereria, कोई भी एक हो सकता है. सेकेंड पे एक्टोपैरासाइट होगा, या तो हेडलाउस या टिक, दोनों में से कुछ तो एक दिखाया जाएगा. एक्सोपैरासाइट, बेडबग या मस्कीटो, कोई एक होगा. एंडोपैरासाइट, एंटामीबा या प्लाज्मोडियम. सो आपको अगर ई पे आता है तो वो एज ए एंडोपैरासाइट लिखना है, एंटामीबा या प्लाज्मोडियम. लेकिन अगर वही ए में आता है तो प्रोटोजोन पैरासाइट के हिसाब से आपको आंसर लिखना है. सो वो ज़रा ध्यान रखो कि कैसे लिखना है. एंड ए टू ई स्पॉटिंग्स आपको करनी है. 10 मार्क्स है, 2 मार्क्स ईच. देर आर 2 मार्क्स फॉर 1 spots. 5th is your submission of BMI report and Viva based on it, which you have already done, and which is for 10 marks. So there are no marks for journal, which is little bit surprising, but it is possible that marks for journal and field report will be also given. So you are supposed to carry field report, BMI report and journal along with you. So this is all about practical 2. So all the best for your exam, and this is all about practical 2 skeleton paper. Thank you.